Okay, so here we are on part 2 of your protein synthesis notes. So part 2 starts off with some differences between DNA and RNA. And the whole reason for this protein synthesis is to make proteins, which is what we just looked at on the front page. You guys are going to see 2 parts that we're going to go through, and you'll do another little hands-on video so you'll see the parts moving around and see how they kind of work together in order to create a protein. The first part that we're going to look at is what's known as transcription. Then we'll go through a process called translation. So what you should know is that chromosomes, which are found within the nucleus that are composed of our DNA, they have our code to make our protein. However, proteins are made out in the cytoplasm. So we somehow have to get this code, DNA's code, from the nucleus out into the cytoplasm where proteins are made. And that's where RNA is going to come in. RNA is kind of like DNA's little brother. So he goes in, he kind of takes that code, but he has to put it in his own language. A couple differences for you between RNA and DNA. So RNA, a couple things about it. It's single-stranded, which means it's smaller. It can fit out of the nucleus, can fit through the nuclear pores a lot easier and make its way to the cytoplasm. It has a different sugar. It has ribose instead of deoxyribose. And one of my nitrogen bases are different. So we said that we have this uracil that's going to replace thymine. Uracil does everything thymine did. It's still a pyrimidine. It's a single ring structure. It will still bond with adenine. It will still have 2 hydrogen bonds with it. And RNA is also found in the nucleus as well as the cytoplasm. DNA can only be found in the nucleus since it's so large. So as we start with this process of protein synthesis, what we need to do is we need to kind of go through our first step, which is transcription. So transcription, what we're going to do is we're going to take DNA and we're going to make it into messenger RNA. Remember I said before, anything that's involving DNA has to take place in the nucleus. I don't care what the process is. If DNA is involved, too big to get out of the nucleus, it's got to take place in the nucleus. So we have 3 types of RNA. We're going to have our messenger RNA, we're going to have transfer RNA, and ribosomal RNA. So mRNA, which is our first type, is going to make a copy. It's going to take DNA's message from the nucleus to the cytoplasm. So it's going to make a copy of DNA. DNA is going to kind of unwrap, and it's going to be used as a template, a mold, or a model in order to make that messenger RNA. But we're only going to be copying a segment of it. We're only going to copy a gene. So it's just a short segment that's going to code for a protein or trait. We're not copying the entire chromosome like we do with replication. Eventually, as messenger RNA is going through and copying that gene, it's going to hit what's known as a stop codon. So a codon is going to be any 3 base pairs in our messenger RNA, and we'll see anticodons when we get to transfer RNA. Remember, any 3 base pairs in DNA is going to be known as a triplet. So at that point, when he reaches what's known as that stop codon that says, hey, this is where the gene ends, messenger RNA breaks off from the DNA strand. My 2 original DNA strands come back together again. Messenger RNA leaves the nucleus, it goes out into the cytoplasm, and it hooks up to a ribosome. This whole process was transcription. When we did replication, we talked about DNA polymerase. Well, now we're going to talk about RNA polymerase. So it kind of does the same job as DNA polymerase, but now we're adding RNA nucleotides. So that's where we're going to be using that. So why do we use RNA polymerase? It's going to bind to DNA during transcription. It's going to assemble or create or make the RNA nucleotides into that complementary messenger RNA strand. So we just talked a little bit about starting and stopping, and we got to kind of know, how does this RNA polymerase know how to do this? Well, there are codes on there that say, hey, this is where my gene starts. It's called a start codon, and this is where I stop. It's called a stop codon. So the enzymes, my RNA polymerase, is going to bind to the promoter region and signal in DNA that shows where polymerase is going to stop at that particular point. We also have areas that are kind of junk, and we talked about this before. Through evolution, our DNA has had invasions from viruses. So viruses come in, they invade our DNA, they become part of our DNA over time, over millions and millions and millions of years. So we have a lot of junk in there within our DNA that we have to kind of cut out. So we have a lot of editing along the way. So when messenger RNA copies DNA, he doesn't want to copy that junk material. So there are things called introns and exons. So I say it's kind of like if you're writing an English paper. The first time you go through, you kind of just throw your ideas down. There may be some stuff that's not going to be kept. That's kind of what those introns are. It's kind of like that rough draft. These are portions that are cut out and they can be discarded a little bit later on. They're the portions that are not going to code for that particular gene. The exons, if you finish up your paper, you cut out all that junk material, you're left with the good draft, the good copy. That's basically your exons. It is that final draft of that English paper. So we're going to splice those segments together and make that final draft. We're going to splice them together and make that final messenger RNA strand that says, hey, this is what's going to code for that protein or trait when I finally get it out to the cytoplasm. Once we're out in the cytoplasm, we're going to utilize some other types of RNA. So my next step is known as translation, and translation is the second step of protein synthesis. And this is where messenger RNA is now turned into proteins. But we're going to utilize transfer RNA, and we're going to utilize ribosomal RNA. And what transfer RNA does, he's kind of like a little taxi cab. He's going to carry messenger or amino acids to the messenger RNA strand. Messenger RNA tells tRNA basically what amino acid to taxi in, to bring into him based on the code that he has. When we get to translation, you will see 3 steps. You'll see it in the diagrams in the next video. You'll also see it on the hands-on activity that I'm going to include a little bit later on today. The 3 steps are going to include initiation, where I'm going to start building my amino acid strands, elongation, where I'm going to grow the strand, and termination, where I'm going to end the strand, saying, hey, that's the end of my protein at that particular point. So there's messenger RNA, there's transfer RNA. We're going, well, okay, what does rRNA do? He's kind of the one that always gets forgotten. And what rRNA does, it's ribosomal RNA. So out in the cytoplasm, you have all these little ribosomes, and they're basically where proteins are synthesized or made. But they're made up of 2 subunits, a large subunit and a small subunit. So our RNA is going to kind of glue them together and activate them so that they are able to go through and read the code for messenger RNA. Once that's done, we can now go through and assemble the polypeptide. So remember, polypeptide is just another name for protein. They're synonyms for each other. This is what we have so far. All my RNAs are made in the nucleus. All the amino acids are in the cytoplasm. They come from the food you eat. And all proteins are made in the cytoplasm. Once those RNAs are made in the nucleus, they're going to travel out to do their job. A gene must be able to direct the order in which the amino acids are put together. So a gene is that segment of DNA within the nucleus that's going to code for the protein or trait. We said on DNA, every 3 base pairs was called a triplet. When we get to messenger RNA, every 3 base pairs is called a codon. So if I think about these codons, I'm going to have 64 possible combinations or ways that I can arrange my base pairs in messenger RNA. So if I take A, U, G, and C, I can arrange them in 64 different ways. AAA, AAU, AAG, AAC, and so on. These are the codes that are going to code for protein synthesis. In tRNA, I have anticodons, and they are 3 base pairs on that transfer RNA. There are only 61 possible transfer RNAs, though, and the reason is because when we get to messenger RNA, we're going to see how it reads for a stop codon, or the 3 stop codons, and transfer RNA doesn't have those particular stop codons, so it will never code for the 3 messenger RNA stop codons. So when we talk about the start and stop code, the start and stop code is going to be found on your messenger RNA strand. So your start codon is always the same. The ribosome must read AUG on the messenger RNA strand. If he doesn't read that, that messenger RNA strand goes right through the ribosome. So he waits until he reads that particular 3 base pair, which is my codon. That is the start of my protein. It actually codes for the amino acid called methionine. So start codon is my step 1. It's called initiation. As it goes through, the ribosome is going to keep going through and reading my start codons. As he does, he's going to call in the appropriate transfer RNAs to match up for it. So for instance, my start codon was AUG. That was the code in messenger RNA. Transfer RNA is going to hook up to AUG. And what's going to happen? A will pair with U will pair with A, G will pair with C. Complementary base pairs. So I will have UAC is going to be the anticodon on my transfer RNA that's going to hook up to my messenger RNA. The other end of my transfer RNA has my amino acid, and we're going to keep growing it this fashion. As I keep growing that polypeptide chain, it's called elongation. Eventually, at the end of my messenger RNA strand, to symbolize that I'm at the end of my protein, or the end of the gene that wants to make that protein, I'm going to hit one of 3 stop codons, UAA, UAG, UGA. As soon as the ribosome reads one of them, it's going to end making that particular protein, and my messenger RNA strand is either going to break down, or if I need more of the same protein, it's going to hook up to another ribosome to make more. My ribosome will break down to its 2 subunits. Transfer RNA keep floating around within your cytoplasm to pick up more amino acids for the next protein being made. So why is this important? Well, we talk about these genes and enzymes, and some scientists went through, and there was a scientist by the name of Gerard who discovered that genes, that segment of DNA, codes for enzyme production. Well, we also know that enzymes are proteins. So he comes up with the hypothesis, 1 gene, 1 enzyme hypothesis. He says that each gene, each segment of DNA, codes for 1 specific enzyme or protein. So those people who are lactose intolerant, you can't break down the milk sugar, your enzyme that your body cannot make is lactase. Somewhere along the line, your little DNA code is messed up and you cannot produce that particular protein. The other hypothesis is the 1 gene, 1 polypeptide hypothesis. It's basically saying the same thing, because we know that a polypeptide is also a protein. So the 1 gene, 1 polypeptide hypothesis says that 1 gene codes for 1 particular protein, kind of like what we looked at with the 1 gene, 1 enzyme hypothesis, since we know that an enzyme is also a protein. Next little video, we'll go through this again within the diagrams. So what I want you guys to do is pull out the diagrams. We're going to start with the one that says transcription. We'll go through transcription, translation, and then it may help you to go through this video again to look at those notes once you see all these particular parts kind of moving around on you. So get out the diagrams. Next video will be on them.